Hi, we're continuing with life science and cell biology in a nutshell. Um, so, animals exploit the phospholipid asymmetry of their plasma membranes of cells. So, asymmetry means the two monolayers of the bilayer are not the same. So, the inner membrane is usually more negatively charged, and there's different kinds of phospholipid molecules in the outer and inner membranes, which help with the specific signaling pathway across membranes of cells. So, phosphatidyl choline and sphingomyelin are two phospholipids in the inner monolayer. Phosphatidyl ethanolamine and phosphatidylserine are in the outer membranes. So, when animal cells undergo programmed cell death or apoptosis, phosphatidylserine, which is usually confined to the cytosolic monolayer of the plasma membrane lipid bilayer, rapidly translates to the extracellular monolayer. What did I say? So, the asymmetry in lipid bilayers is generated and maintained by membrane-bound phospholipid translocators, which are like enzymes or proteins.
What's the sip of water?
So phosphatidylserine is in the inner membrane. It creates a negatively charged inner membrane. So phosphatidylcholine and sphingomyelin are in the outer membrane. I think I said it backwards earlier. And phosphatidylethanolamine and phosphatidylserine are in the inner monolayer. So as shown here in the diagram, the first two with terminal amine group are in the inner membrane. The second two are in the outer membrane. So when phosphatidylserine goes to the outer cell surface, it is exposed to the outer um, outside of the cell. It's serving as a signal to induce neighboring cells such as macrophages to phagocytose the dead cell and to digest it. So this only happens in the situation when the cell is going to be recycled. The translocation of the phosphatidylserine in apoptotic cells occurs in the two mechanisms. The phospholipid translocator, normally transporting the lipid from the non-cytosolic monolayer to the cytosolic monolayer, is inactivated, and to a scramblase that transfers phospholipids non-specifically in both directions between the two monolayers is activated. Glycolipids um, are a molecule found in the plasma membrane. The lipid molecules with the most extreme asymmetry in their membrane distribution are the sugar-containing lipid molecules called glycolipids. These intriguing molecules are found exclusively in the non-cytosolic monolayer of the lipid bilayer, where they're thought to partition preferentially into lipid rafts, which we mentioned earlier. The glycolipids tend to self-associate partly through hydrogen bonds between their sugars and partly through van der Waals forces between their long and mainly saturated hydrocarbon chains. The asymmetric distribution of glycolipids in the bilayer results from the addition of sugar groups to the lipid molecules in the lumen of the Golgi apparatus, which is topologically equivalent to the exterior of the cell. In the plasma membrane, the sugar groups are exposed as at the cell surface, where they have important roles in interactions of the cell with its surroundings. Glycolipids probably occur in all animal cell plasma membranes, where they're generally constituting about 5% of the lipid molecules in the outer monolayer. They are also found in some intracellular membranes inside the cell. The most complex of glycolipids, the gangliosides, contain oligosaccharides with one or more sialic acid residues, which give gangliosides a net negative charge. So diagram showing what these look like. A glycolipid molecule. So a cerebroside is called a neutral glycolipid because the sugar that forms its head group is uncharged. A ganglioside always contains one or more negatively charged sialic acid residues, also called N-acetyl-neuraminic acid or NANA, whose structure is shown over here. Whereas in bacteria and plants, almost all glycolipids are derived from glycerol, as are most phospholipids. In animal cells, however, they are almost always produced from serine, as in the case for the phospholipid sphingomyelin. So in this diagram, GAL, galactose, GLC, glucose, GAL-NAC is N-acetyl-galactosamine. So these three sugars are uncharged, and they are bound to the phospholipid glycerol and fatty acid chains, making up the glycolipid. So the first glycolipid is neutral. The second one is the negative. It contains a negatively charged acetyl-neuraminic acid, NANA, which is also called sialic acid. So this particular sugar has the carboxyl group COO, making it negatively charged. So it contains a ring structure, 6-carbon ring. So carbons, oxygen, hydrogen, and nitrogen. And the sugar, more than 40 different gangliosides have been identified. They are most abundant in plasma membrane of nerve cells, where gangliosides constitute 5 to 10% of the total lipid mass. They are also found in much smaller quantities in other cell types besides just nerve cells. So these are lipid and sugar glycolipids. Hints as to what the functions of glycolipids might be come from their localization. In the plasma membrane of epithelial cells, for example, glycolipids are confined to the exposed apical surface, where they help to protect the membrane against the harsh conditions frequently found there, such as low pH and degradative enzymes. Charged glycolipids, such as gangliosides, may be important for the electrical effects. Their presence alters the electrical field across the membrane, and the concentrations of ions, especially calcium 2+, at the membrane surface. Glycolipids